Hello. Good evening to the students who have joined. Can you please let me know in the chat box if my voice is audible to you? And also please let me know if you can see the presentation clearly. Good evening to the students who have joined. Please let me know if my voice is audible to you, and also please let me know if you can see the presentation clearly. Am I audible? Okay, thank you for confirming. We will be starting the session from 6 p.m. So please wait till then. Let other students join. Then we will start the session. If there is any doubt from the last class or any of the last classes, you can let me know now. Okay, so I think I will be starting the session by introducing myself. But before that, I hope you all know already that where to find the class material, whatever I am showing today, and the class recordings and all are available in the SWAM portal in the course page. If you go to the announcement page, you will find there is a on the left panel there is a problem solving session tab. If you click on that tab, you will be finding two options. One is the problem solving session recording and other is the survey. So if you click on that problem solving session recording, you will be redirected to a page which okay, yeah. If you click on here, you will be redirected to a page which looks like this. Okay, and here you will be finding the particular YouTube links for every class recording, and also you will be finding a Google Drive link. This Google Drive contains all the presentation which I show during our class. Okay, so there from here you can access the materials whatever we are doing in the problem solving session. All materials are available in this page here. Okay, so it's 6 p.m. I think I will be starting the session. Okay, I hope my voice is audible and you can see the presentation clearly. If not, please let me know in the chat box. Okay, let me start by introducing myself. So, very good evening to the students who have joined. My name is Mousumi Deb and I'm PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 5 live session. Before starting the session, let us discuss few points. So there will be this kind of problem solving session every week, and my classes will be on every Monday from 6 to 8 p.m. This is a 12-week course, so we will be having 12 consecutive live sessions on Mondays from 6 to 8 p.m. and one summary session. Regarding that summary session, the schedule will be shared to you at the end of the course. Okay, the summary session will be also of 2-hour session, and it will contain all your course content in a summarized form before your examination. Okay, so all the sessions are live on Google Meet platform. The link is same for every Monday class. This link will be the same throughout our session. Okay, so if any network disruption happens from my side, do not leave the session immediately. I will try to rejoin as soon as possible, and during the session you can post your doubts and queries through the chat box. Since I'm recording the session and so that I can record the session without any problem, I have muted your microphones and cameras, but the chat box is always open and I am monitoring the chat box every now and then. Okay, the chat box will be the media for our communication throughout the session, so please use the chat box for answering the questions and asking your doubts and queries. Okay, after the session, if the doubt persists, you can drop an email to me. I will try to answer your query via email also, and if it is not possible to do the same, I will try to answer your query in the upcoming class. Okay, so after today's class, if there is any doubt, any query regarding today's class or any previous classes, please let me know in the class or after class via email or through discussion forum. I will try to discuss that particular topic in the upcoming classes. Okay, so as I have mentioned, this presentation and class recording will be shared to you, and where to find the class recordings and where to find the presentation I have already shown you. Okay, so you can download the class materials and see the recording after the class. Okay, generally I try to upload all the things by the same day, or it will be available to you by tomorrow. Okay, so let us start today's class. So today it is a week 5 live session. So in every live session, what we do, we discuss that particular week's course content. Since it is a week 5, so today we will be discussing the week 5 course content. Okay, so in the week 5, let us first see what are the topics of the week 5 course content. So we have the nucleic acids as the drug target. Then we have miscellaneous drug targets and related topics with nucleic acid, and little bit we will be discussing about the various mechanisms of the biological chemistry. So in our last class, what we have discussed, so in the last session we have this in the class session 4, we have discussed about different receptors as the drug target, but today we will be focusing on the nucleic acids and the enzymes which are related to the nucleic acid as the various drug targets. And we have already discussed about the importance of stereochemistry and all these things in the last session. Okay, so today our topics will be like this nucleic acids and the related topics. Okay, so like we do in every class, I will be showing you different MCQ questions. All the questions will be having for different answer, and after showing you a particular question, I will wait for 1 to 2 minutes and then I will check the chat box. You have to let me know A, B, C or D which of the following will be the correct answer for the problem, and then I will check the chat box, check your answers, and then I will show you the correct answer, and we will discuss the topic in detail. I will discuss the topic in a such a manner that if there is a subjective questions are asked in the final examination, because in your assignments the MCQs are only asked, so that is why we have designed the problem solving session like for as only with the MCQs, but all the questions are comes up with every very detailed answer. So if there is any subjective answer question are asked for you in the final examination, you will be it will be easier for you to answer the questions. Okay, so let us start with the first question of today's session. So the first question of today's session, it says that identify the incorrect statement about camptothecin. Your options are: number A, it inhibits the DNA polymerase enzyme; number B, it is a naturally occurring cytotoxic quinoline alkaloid; number C, its lactone form is active; and number D, it triggers apoptosis. Which of the following statements regarding camptothecin is incorrect? Again, I will repeat the options. Number A, it inhibits the DNA polymerase enzyme. Number B, it is a naturally occurring cytotoxic quinoline alkaloid. Number C, its lactone form is active. And number D, it triggers apoptosis. Which of the following statement is incorrect regarding camptothecin? Let me know in the chat box A, B, C or D, which of the following you think is the incorrect statement regarding camptothecin. So whenever there is this type of correct, incorrect, true, false type of question, please read the options very carefully and then answer, because most of the students have the tendency to make a mistake in this kind of questions, because sometimes the options can be a little tricky and a little confusing. So think carefully and then let me know. The statement which is incorrect about camptothecin. I will wait for a minute, then I will check the chat box. It inhibits the DNA polymerase enzyme. Number A. Number B, it is naturally occurring cytotoxic quinoline alkaloid. Number C, its lactone form is active. And number D, it triggers apoptosis. Okay, let me check the chat box. Let's see if there is any answer for the first question. Okay. So I have received two answers. One is saying number D is the correct answer and another is saying number C. Let me check which of the following is the correct answer for problem number 1. So the correct answer is number A. It inhibits the DNA polymerase enzyme. This statement is incorrect about camptothecin. Now let us look what is camptothecin and what is it all about. So basically camptothecin is a cytotoxic quinoline based plant alkaloid. Okay, and it is most established inhibitor of DNA polymerase 1. So the first option which says that it inhibits the DNA polymerase enzyme, this is false, okay, because it is the inhibitor of DNA topoisomerase 1 enzyme. Topoisomerase 1 is the only target of this drug, okay. So this drug is very important and very established drug of inhibitor of topoisomerase 1, because it is the only target for this drug. It does not show any kind of cross target activity or so, or any kind of multi target activity. So it is a very important and crucial drug which take uses this particular enzyme DNA polymerase. Okay, DNA topoisomerase. CTP binds binds neither to top 1 alone or not to the DNA alone, but only to the top 1 DNA cleavable complex. Now, first, let us understand what is the role of the topoisomerase. I think I have discussed it in our last class. We had discussed about the topoisomerase. Okay, so topoisomerase is the enzyme. So basically, whenever there is a replication, the double helix DNA needs to be the double helix of DNA. It needs to be formed a single strands, okay, so that the replication can happen. So whenever there is this unwinding of the double helix, there is a torsion that is created at the position of the unwinding, okay. And what happens at the position of the torsion? So this topoisomerase enzymes, they basically relieves that particular torsion. So what does they do? They bind to the DNA and they relieve the torsion by unwinding. The mechanism, whatever, how the topoisomerase work, the topoisomerase 1 and 2 already I have shown you. You can go through that particular slide. I think it is in the week 2 or week 3. I do not remember correct. I mean exactly, but it is there. Okay, and it relieves the particular. This particular. A torsion or tension which is created in that position, but neither this particular but this topoisomerase this camptothecin, it acts as the particular inhibitor of DNA topoisomerase 1, but it does not bind to the enzyme single handedly, okay. It only binds when this topoisomerase is bound with the DNA, okay. And that is known as the cleavable complex or topoisomerase DNA cleavable complex. So camptothecin is the naturally occurring cytotoxic quinoline alkaloid, first isolated in 1966 from the bark and stem wood of the Chinese tree Camptothecea acuminata, or also known as the happy tree. It is one of the most significant natural anti-cancer agent alongside the Taxol and Vinca alkaloid. Okay, the mechanism of action: camptothecin is a potent inhibitor of DNA topoisomerase 1, an enzyme essential for relieving DNA supercoiling during the replication and transcription. Tertiary complex CPT. This camptothecin is also known as the CPT in the short form, the abbreviated form, okay. CPT binds superficially, specifically to the transient topoisomerase 1 DNA cleavage complex, forming a stable ternary complex that prevents the enzyme from religating the DNA. Replication fork collision during the S phase of the cell cycle, advancing replication fork collide with this stabilized complex, causing irreversible double-strand DNA breaks. Apoptosis, the resulting DNA damage, triggers the programmed cell death or apoptosis primarily through the mitochondrial intrinsic pathway, okay. And this is the structure of the particular camptothecin. This is the SAR, the structural activity relationships. As you can see, there are 5 rings which are conjugated together, okay. So this this is a this is a planar structure, and the rings are named as A, B, C, D, and E. So as you can see, C and D ring modification leads to the loss of the cytotoxicity. So these positions cannot be modified. Modification of the ring A and B are well tolerated. The saturation of the ring A and B lowers the activity. Okay, the D ring, this pyridone structure is essential for the anti-cancer activity. Hydrolysis of this E row leads to the inactive carboxylate form. This C double bond O is necessary for the activity. C double bond N and C double bond S is inactive in this position. C20 S isomer is inactive and R isomer is inactive. So we have already seen how the activity is depends on the particular stereochemistry. In our last class we have discussed. So in this case, this lactone ring is active, okay. The lactone formation of this camptothecin is active, but if this lactone E ring get hydrolyzed and form carboxylate, this is the inactive form. So now let us see our options. The first option says it inhibits the DNA polymerase enzyme. This is incorrect because it inhibits the DNA topoisomerase enzyme. It is a naturally occurring cytotoxic quinoline alkaloid. This is true. Its lactone form is active. This is also true. It triggers apoptosis. This option is also true. So the incorrect option is number A. So is it clear, or do I need to repeat that question again? Okay, if it is clear, then it's fine. Otherwise, I will. So if you have any doubt or any query, please let me know. I will repeat that particular question. Okay, if you get any doubt or any problem. If there is no doubt, then I will move toward our next problem. So problem number 2 says, which of the following is a semi-synthetic derivative of camptothecin? Your options are number A, irinotecan, number B, topotecan, number B, velotecan, and number D, all of the above. Which of the following is a semi-synthetic derivative of camptothecin? Irinotecan, topotecan, velotecan, or all of the above? Let me know A, B, C, or D. Which of the following will be the correct answer of problem number 2? Which of the following is a semi-synthetic derivative of camptothecin? Irinotecan, topotecan, velotecan, or all of the above? Again, I will wait for a minute, then I will check the chat box. See, when the nucleic acid is the drug target, there are multiple mechanisms or through which this particular drugs can operate. Okay, I will try to give you at least one example of each and every class, and whatever is remaining, we will discuss in our summary session. Okay, let me know in the chat box A, B, C, or D which of the following you think is correct answer for problem number 2. Which of the following is the semi-synthetic derivative of camptothecin? I will wait for a minute, then I will check the chat box. Which of the following is a semi-synthetic derivative of camptothecin? Let me check the chat box. I hope I will find some answers there. So I have received only one answer, which is saying number B is correct. Others are not answering. Okay, let me check if number B is the correct answer or is there any other answer which is correct. So the correct answer, the derivative which is a semi-synthetic derivative of camptothecin will be, the correct answer is number D, all of the above. Okay, so irinotecan, topotecan, velotecan, all of the above are the examples of the semi-synthetic derivative of the camptothecin. So the clinical status and FDA approved derivatives, while pure camptothecin showed impressive anti-tumor activity in early trials, its clinical use was halted in 1970s due to the extreme water insolubility, instability of the active lactone ring at the physiological pH, and severe toxicities like hemorrhagic cystasis and myelosuppression. To overcome this, several semi-synthetic analogs have been approved by FDA. For example, this is irinotecan, which is also known as CPT11, a water-soluble prodrug primarily used for metastatic colorectal cancer. So this is the structure of irinotecan. As you can see, the modification. So the basic structure is same. The basic structure of camptothecin remains same. Other there are little bit of modifications are there in this A and B ring. Okay. Then we have this. Then we have the topotecan, or also it known as the hycamtin. So this is the topotecan approved for the second line treatment of the small cell lung cancer, ovarian cancer, and cervical cancer. Then we have this trastuzumab deruxtecan, then antibody drug conjugate or ADC using a CPT derivative as the cytotoxic payload to target the HER2 positive cancers. And then we have this sacituzumab govitecan, which is also antibody drug conjugate targeting TROP2 expressing tumors such as triple negative breast cancer. And lastly, we have the belotecan, which is this one approved in South Korea but not currently by US FDA for the lung and ovarian cancers. Okay, so the correct answer was number D. All of the above are the semi-synthetic derivatives of camptothecin. So the structure of camptothecin, the structure, you have to remember the particular structure of camptothecin. Okay, it is important very much because also the SAR is very important of the camptothecin. By the SAR means the parts of the camptothecin, the rings, where the changes can be done, where the changes are not tolerated, and the lactone ring. So all these things which I have discussed in the first question, this part is very important. You have to remember, and also this examples, the structures. If you can remember, it is very good, well and good. If not, at least remember the names of the derivatives. Okay. So I hope it is clear. If there is any doubt, please let me know via chat box. Okay. If there is any query or any doubt regarding any question, please feel free to let me know. Okay, now I will go to the next problem. So problem number 3, it says that What is the mode of action of proflavin? Your options are number A, alkylating agent, number B, chain cutting agents, number C, antisense agents, and number D, intercalating agents. What is the mode of action for proflavin? Alkylating agent, chain cutting agent, antisense agent, or intercalating agents? Let me know. A, B, C, or D, which of the following will be the correct answer of problem number 3? Again, I will be waiting for a minute or two and then I will check the chat box. Which of the following is the correct mode of action for proflavin? Number A, alkylating agent. Number B, chain cutting agent. Number C, antisense agent. Number D, intercalating agent. Which of the following agents proflavin behave? Let me know. Whatever you think is the correct answer, let me know via chat box. There is no marking, there is no negative marking for answering during the class, but please try to answer the questions. Otherwise, I won't understand if you are following the class or not. Okay? Whatever you think is the correct answer, let me know. Proflavin works as which kind of agent? Alkylating agent, chain cutting agent, antisense agent, or intercalating agent? What will be the correct answer? A, B, C, or D? What is the correct mode of action of proflavin? Let me check the chat box. Let's see if there is any answer. Okay, I have received a couple of answers. Number D, number B, number C. Okay, so I have received three different options as answer. Number D, number B, and number C. So let me check which of the following will be the correct answer, or is there any different answer which is correct? So the correct answer will be proflavin works as number D, intercalating agents. Okay. So this is the structure of proflavin. So it is a topical antiseptic agent used in the wound dressing to prevent infections. Okay. So this is the drug which is an example for the intercalating agent. Now what is an intercalating agent? So intercalating agents, they contain a flat planar structure that intercalate themselves into the double helix of the DNA. So this ring, as you can see, it is a planar structure which is flat, which can fit into the double helix of the DNA like this, and it can interact with this nucleobases with the van der Waals interactions. Okay, and there is also a polar side part, which is this ammonium ion, which actually interacts with this sugar phosphate backbone with a with the this ionic interactions. Okay, so so this interactions and this intercalation distorts the double helix structure. At replication, a template that contains an intercalated agent will cause insertion of random extra base pairs. The base pair addition is complete after one round of replication and during which the intercalating agent is lost. For example, ethidium bromide, proflavin, acridine, orange, all are the examples of the intercalating agents. So intercalating drugs are compounds that contain a planar heteroaromatic features which slips between the base pair layers of the DNA double helix. Some of these drugs prefer to approach the helix via the major group, other prefers to access via minor group. Once they are inserted between the nucleic acid base pairs, the aromatic heteroaromatic rings are held there by van der Waals interaction with the base pairs above and below. Several intercalating drugs also contain ionized group which can interact with the charged phosphate groups of the DNA backbone, thus strengthening the interaction. Proflavin is an example of the group of antibacterial compounds called as the aminoacridins, which were used during the World War I and II to treat the deep surface wounds. These drugs proved highly effective in preventing infection and reduce the number of fatalities resulting from wound infection. Proflavin is completely ionized at pH 7 and interacts directly with the bacterial DNA. The flat tricyclic ring, this one, intercalates between the DNA base pairs, okay, and interacts with them with the van der Waals forces, while the ammonium cations form the ionic bond with the negatively charged phosphate group of the sugar phosphate backbone. Once inserted, proflavin deforms the DNA double helix, prevents the normal functions of replication and transcription. Okay, so this is the basic, this is the common, mechanism of actions for the intercalating agents. Proflavin is only an example. Other examples are ethidium bromide, actinium orange, which also acts as the intercalating agents. Okay, so try to remember the structure of proflavin, how the mechanism of intercalation happens. So these are the important topics. Okay, so is there any doubt? Otherwise, I will move to the next problem. Please let me know if there is any doubt. If not, then I will move to the next problem of today's session. So problem number 4, it says that tubulin polymerization inhibits the trigger tubulin polymerization inhibitors triggers cell cycle arrest during dash phase. Your options are number A S phase, number B G1 S phase, number C G2 M phase, and number D M phase. Tubulin polymerization inhibitors trigger cell cycle arrest during which phase? S phase, G1 slash S phase, G2 slash M phase, or M phase? Let me know A, B, C, or D. Which of the following will be the correct answer? Tubulin polymerization inhibitors trigger cell cycle arrest during which phase? I will wait for 1 minute and then I will check the chat box. Okay. Let me know A, B, C, or D. What do you think? What is the correct answer? Tubulin polymerization inhibitors trigger cell cycle arrest during dash phase. What will be the correct answer? Let me check the chat box. Okay. So I have received one, two, number C, number A, number A, number C, number C. So I have basically received two different options, number C and then number A as the answers. Okay. Let me check which of the following will be the correct answer, number A or number C. So the correct answer will be the tubulin polymerization inhibitors trigger cell cycle arrest during G2M phase. Okay, C was the correct answer. Now let us first see what is the cell cycle. So this I will also discuss later in our course when we will be discussing solely about the anti-cancer agents. So this is the cell cycle. Okay, so during a cell division, the cell particularly goes through different cycles, so called different phases. First one is the G1 or cell growth phase, where the cell prepares itself for the particular replication. Okay, so the cell division. So if the after the G1 phase the cell is not prepared, so it will go to the G0 resting phase and then resume the cell cycle after it gets all the important nutrients. Then it goes to the the replication or S phase. After that we have this G2 phase, which is a preparation for mitosis page, and then M phase. M phase, their mitosis happens, which is the prophase, metaphase, anaphase, and telophase for the different phases of the cell division, and then cytokinesis happens. Okay, so this is the cell division: cell growth, DNA replication, preparation of mitosis, and cell division. So this is the complete cell cycle. Okay, so tubulin polymerization inhibitors are the major class of the microtubule destabilizing agent that interfere with the assembly of alpha beta tubulin heterodimers. By blocking the formation of the mitotic spindle, these agents trigger the cell cycle arrest as the G2M phase, ultimately leading to the programmed cell death or apoptosis. What is the mechanism of action? They disrupt the dynamic instability of microtubules required for the chromosome secretion during the mitosis. Okay, so during the mitosis, the dynamics instability of microtubules is required for chromosome secretion. Okay, so is disrupted by these inhibitors binding to free tubule. Most this tubulin polymerization inhibitor bind to the unpolymerized tubule dimer, preventing them from being added to the growing end of the microtubule. Depolymerization at higher concentration, they actively promote the disassembly of the existing microtubule polymer, decreasing the total microtubule mass in the cell. Major binding domains and agents. So there are different categories of this tubulin specific inhibitors based on the specific binding pockets on the tubulin protein. So when the binding site is the vinca site, the key agents are the vincristine, vinblastine, and vinrolibin. So they are a long-standing FDA approved mainstays for the lymphomas, lung, and breast cancer. Colchicine site, colchicine, combristatin A to A4. There are no colchicine site inhibitor is currently FDA approved for cancer due to toxicity, but many are in the phase 2 or 3 clinical trial. Mastine site, so for example, are the martensine and raftensin. So they primarily utilized as high potency payloads in the antibody drug conjugates. Other for like eribulin, so they are FDA approved for the metastasis breast cancer and liposarcoma. It binds near the vinca site. Okay. So these are the drugs which actually targets the tubule. Vinca alkaloid, vincristine, vinblastine, they binds with the tubulin and inhibits the microtubule formation, and it goes to the cell, goes to the metaphase arrest, and it acts as an anti-cancer agent. We have the taxanes, which are also used as an anti-cancer agent. They actually freezes the microtubule in metaphase, and no anaphase stage happens here. Colchicine binds to tubulin, inhibit the microtubule polymerization, impair activation and no migration. Okay, so it's used for the neutrophils or the for reducing the inflammation in gut. Then we have the grisfolvin, which binds to the fungal tubulin, impaired microtubule formation, inhibit mitosis. These are acts as a fungistatic agents. Okay, and then we have the mebendazole or albendazole. So they block the polymerization of tubulin and in the intestinal cells, blocking the glucose and glucose uptake, gradual immobilization and death of the hermesis. So these are the different tubulin targeting drugs. Okay. So this was the problem number 4. Now I will go to the next problem. Problem number 5 says doxorubicin is an intercalating agent which belongs to a group of naturally occurring antibiotics. What role does the ionic group serve to enhance the intercalation effect of this molecule? Your options are number A, it increases the water solubility allowing high dose to be used. Number B, it interacts with the charged phosphate group in DNA to stabilize the intercalation. Number C, it acts as a strong hydrogen bond donor with the nucleic acid bases to stabilize the intercalation. Number D, it only becomes protonated when it enters the cell, so it becomes trapped within the cell, leading to high concentration of the drug. Which of the following is the correct answer for problem number 5? Again, I will repeat it one more time. Doxorubicin is an intercalating agent which belongs to a group of naturally occurring antibiotics. What role does the ionic group serve to enhance the intercalation effect of this molecule? Your options are number A, it increases the water solubility, allowing high dose levels to be used. Number B, it interacts with charged phosphate group in the DNA to stabilize the intercalation. Number C, it acts as a strong hydrogen bond donor with the nucleic acid bases to stabilize the intercalation. And number D, it only becomes protonated when it enters the cell, so becomes trapped within the cell, leading to the high concentration of the drug. Again, I will wait for a minute, read all the options carefully, and then let me know A, B, C, or D, which of the following will be the correct answer for problem number 5. What role does the ionic group serve to enhance the intercalation effect of doxorubicin? Doxorubicin is an intercalating agent which belongs to a group of naturally occurring antibiotics. What role does the ionic group serve to enhance the intercalation effect of this molecule? Let me check if there is any answers for our problem number 5. Okay, number D, number A. So I have received three answers. One is saying number A and other two is are saying that number D is the correct answer. Let me check which of the following is the correct answer for this problem number 5. So the correct answer will be, it is number B. It interacts with the charged phosphate groups in the DNA to stabilize the interaction. Now, just look at the last question, last to last question which we have discussed regarding the proflavin. So when I was discussing the proflavin, I have said that the no common, this is the common mechanism of action for any intercalating drug, that the charged, the polar group, they interact with the sugar phosphate backbone to stabilize the intercalation. The question is this remains same, only the question is framed in that way that you might have the confusions. Okay, so the doxorubicin acts as a similar way. The charged ionic group will interact with the charged phosphate group in the DNA to stabilize the intercalation like any other intercalating agents, for example proflavin, which we have already discussed. Okay, so this is the structure of doxorubicin. As you can see here also there is a ammonium ion just similar to the proflavin. So doxorubicin, okay, so doxorubicin is one of the very most effective anti-cancer drug ever discovered and belongs to the group of naturally occurring antibiotics called the anthracyclines. Okay, so as you can see, this is the structure of the doxorubicin. So this is a structure is almost similar. We have a planar flat structure which can interact with the base double helix of the DNA. It can slide through this double helix, and this flat planar ring can interact with the van der Waal interaction, and the ionic group, it can interact with the sugar phosphate backbone. It was first isolated from the Streptomyces pituosus in 1967. It contains a tetracyclic system where three of the rings are planar. Okay, three of the rings are planar here. The charged amino group attached to the sugar is also important as it forms the ionic bond with the negatively charged phosphate group of the DNA backbone. This is supported by the fact that the structure lacking amino sugar have the pore activity. So if you can see, there is a whenever there is a modifications of doxorubicin is made without this particular group, so you will see there is a pore activity which supports this fact that these ionic bonds are very much, these ionic groups are very much important. Agents such as doxorubicin are referred as the topoisomerase 2 poison rather than inhibitors, as they do not prevent the enzyme function directly. Intercalation prevents the normal action of the enzyme called topoisomerase 2, an enzyme that is crucial for replication and mitosis. Okay, the function of topoisomerase 2 involves the formation of DNA enzyme complex where the enzyme is covalently linked to the DNA. When doxorubicin is intercalated with DNA, it stabilizes this DNA enzyme complex and stalls the process. Okay, but this is known as the topoisomerase 2 poisons rather than inhibitor, as they do not prevent the enzyme functioning directly. So this is similar to what we have discussed regarding the camptothecin. Okay, camptothecin also works as a poison. It does not inhibit the particular topoisomerase enzyme directly, but it stabilizes the DNA enzyme complex. Okay, and that's why these agents which does not stabilizes this DNA topoisomerase complex, these are known as a topoisomerase poison rather than the inhibitor. Okay, inhibitor are the compounds or the drugs which inhibits the enzymes directly, but doxorubicin and camptothecin, both they do not interact with the enzyme directly, but they interact with the DNA enzyme complex and stabilizes this. Doxorubicin does it with the topoisomerase 2 topoisomerase 2 complex, and camptothecin does it with the DNA topoisomerase 1 complex. Okay, so these are also known as the poisons. Okay, but the mechanism of action is the intercalation basically for in case of the doxorubicin. Okay, now I will go to the next problem. Problem number 6 says, Which DNA base interacts with the anti-cancer intercalating agent dactinomycin? Your options are number A adenine, number B thymine, number C cytosine and number D guanine. Which DNA base interacts with the anti-cancer intercalating agent dactinomycin? Adenine, thymine, cytosine or guanine? Let me know A, B, C or D which of the following you think is the correct answer for problem number 6. Which DNA base interacts with the anti-cancer intercalating agent dactinomycin, adenine, thymine, cytosine and guanine? Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C or D which will be the correct answer. Which DNA base interacts with the anti-cancer intercalating agent dactinomycin? Adenine, thymine, cytosine or guanine? Okay, let me check if there is any answers in the chat box for problem number 6. Okay, number C, number D, and I think this is okay. I have received only two answers. Both are saying that number C is the correct one. Let me check if number C is the correct answer or not. So the DNA base that interacts with the anti-cancer intercalating agent dactinomycin, it will be the correct answer is number D, guanine. Okay, so guanine is the base which interacts with the dactinomycin. So this is the structure of dactinomycin. Okay, so another example of intercalating agent is dactinomycin. It previously called as the actinomycin D. It is a naturally occurring antibiotic. First it is isolated from this Streptomyces parvulis in 1953, and it has been shown to have an effect as an effective anti-cancer agent in childhood cancers. If you look into the structure, you will say it has a planar phenoxazone system, which is has a peptide over the cyclic, which has also has a peptide over here, which is a cyclic peptide. So as you can see, this is the phenoxazone planar ring, and we have a cyclic peptide over here. And this drug, as the drug approaches the DNA via the major groove of the double helix, and after it intercalates, the planar tricyclic system gets between two base pairs, which we have seen previously. Okay, the charged amino group attached with the sugar is also important because it forms an ionic bond with the negatively charged phosphate group with the DNA phosphate backbone. It appears to favor interaction with the guanine cytosine base pair or CG base pair. Okay, in particular two adjacent so let me highlight this portion. It appears to favor interaction with the guanine cytosine base pair, that is the GC base pair, and in particular between two adjacent guanine bases on the alternative strands of the helix. So if you have GC interactions and if you have two Gs, then it prefers to bind to this type of DNA base pair. Okay, so basically it prefers the GC base pair, but when you have, if you have two guanine together, so then it will prefer this type of base pair. This molecule is further held in position by the hydrogen bond interaction between the nucleic acid bases and the cyclic pentamers. Okay, the two amino group of the guanine plays a particularly important role in the interaction. The resulting bound complex is very stable and prevents the unwinding of double helix. So in turn, it prevents DNA dependent RNA polymerase from catalyzing the synthesis of mRNA, and it prevents the transcription. So the effect of dactinomycin is actively dividing cell is once it gets in, it goes inside and does the intercalate. After the intercalation, it forms extremely stable bond with DNA, and after that the complex form, because the complex is stable, it prevents the unwinding of double helix, and if unwinding of double helix does not happen, so the single strand DNA will not be produced, which is going to transfer the information to the mRNA. So therefore the transcription is inhibited, and if the transcription is inhibited, then the cell will stop replicating or dividing. Okay, because the transcription or the protein synthesis is a very crucial step of the central dogma of the molecular biology. Okay. So this is the mechanism of action for the dactinomycin, and remember that it prefers the particular DNA-based guanine and also guanine cytosine base pairs it preferred. But the highest, I mean, preference is the highest preference is to the with the guanine. Now I will go to the next problem. Problem number 7 says, which enzyme is responsible for the metabolism of cyclophosphoramide? Your options are number A, cytochrome P450, number B, cytochrome C oxidase, number C aconitase, and number D ligase. Which enzyme is responsible for the metabolism of cyclophosphoramide? Your options are number A, cytochrome P450, number B, cytochrome C oxidase, number C aconitase, and number D ligase. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 7. Again, I will wait for a minute or two, then I will check the chat box. A, B, C or D, which of the following will be the correct answer for problem number 7? Which enzyme is responsible for the metabolism of cyclophosphamide? Cytochrome P450, cytochrome C oxidase, aconitase or ligase? Which enzyme is responsible for the metabolism of cyclophosphamide? Cytochrome P450, cytochrome C oxidase, aconitase or ligase? Which enzyme is responsible for metabolism of cyclophosphamide? Let me check the chat box. Okay, number A, number A, number A. So basically, I have received 3 answers and all are saying that number A is the correct answer. Okay, let me check if number A is correct answer or not. So the correct answer will be yes, it is number A. Cytochrome P450 is the enzyme which is responsible for the metabolism of cyclophosphamide. Now let us see what is the mechanism of action. So cyclophosphamide is the most commonly used alkylating agent in the cancer chemotherapy. So now this we have got another different kind of agent. Previously we were discussing about the intercalating agent. Now we have a agent which is known as the alkylating agent, and cyclophosphamide is a very good example for the alkylating agent in the cancer chemotherapy. Okay. So it is a non-toxic prodrug which is converted to the body by the active drug. What is a prodrug and what is the necessary of prodrug we will be discussing in our upcoming classes. Metabolism takes place in the liver where cytochrome P450 enzymes oxidize the ring. Ring opening then takes place and a non-enzymatic hydrolysis splits the acrolein from the molecule to generate the cytotoxic alkylating agent. So this is the cyclophosphamide, okay, and then cytochrome P450 enzymes, they basically adds a hydroxyl group in this position. After that, the hydrolysis takes place and the cytotoxic alkylating agent it is formed. Okay, and another side product which is formed is acrolein molecule. So the nucleophilicity of the nitrogen is tempered by being a part of the phosphoramide group, like here, and so the active agent is more selective for the stronger nucleophile such as the guanine. So this is also important to remember. Okay. The active cytotoxic alkylating agent is selective for the stronger nucleophiles such as the guanine. Cyclophosphamide itself is relatively non-toxic and can be taken orally without causing damage to the gut wall. It was Also hope that the high level of phosphoramide this enzyme present in some tumor cell will lead to the greater concentration of alkylating agent in this cell and result in some selectivity of action. Unfortunately, the acrolein released, this acrolein sometime can proven to be toxic to kidneys and bladder. One possible explanation is the acrolein alkylates the cysteine residues of the cell protein. So, certainly, toxicity can be reduced by co-administrating with the sulfhydryl donors such as the N-acetylcysteine or sodium 2 mercaptophenone sulfonate which interact with the acrolein. So these are the co-administrating drugs which are necessary to remove the toxic action of the acrolein, which is formed when this prodrug cyclophosphoramide is converted to the active cytotoxic alkylating agent. Okay. So this is very important. Okay, this cyclophosphoramide and the mechanism of action of the cyclophosphoramide, how it behaves. So this is very important. And the mechanism of action, this for the same synthesis of the phosphoramide mustard from the yes, this cyclotoxic alkylating agent also known as the phosphoramide mustard. Okay, so the this conversion is very important to practice it. I think it is not very difficult. You will be certainly do it perfectly. Okay. Now I will go to the next problem. Problem number 8 says etoposide is number A intercalating topoisomerase poison, number B non-intercalating topoisomerase poison, number C intercalating topoisomerase inhibitor, and number D non-intercalating topoisomerase inhibitor. This is the structure of the etoposide. You have to identify what kind of agent it is. Number A, intercalating topoisomerase poison. Number B, non-intercalating topoisomerase poison. Number C, intercalating topoisomerase inhibitor. And number D, non-intercalating topoisomerase inhibitor. Let me wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer. Etoposide behaves as which kind of reagent? Intercalating topoisomerase poison, non-intercalating topoisomerase poison, intercalating topoisomerase inhibitor, or non-intercalating topoisomerase inhibitor. Etoposide is intercalating topoisomerase poison, non-intercalating topoisomerase poison, intercalating topoisomerase inhibitor, or non-intercalating topoisomerase inhibitor. Let me check the chat box. I think number D, number C, number C. So I have received three different and three answers, two number C and one number D. Let me check which of the following options will be correct. So the correct answer will be, it is number B, non-intercalating topoisomerase poison. Okay, so etoposide is a non-intercalating topoisomerase poison. So non-intercalating topoisomerase poison, the anti-cancer agent etoposide and teniposide, they belong to a class of compound which are known as the podophylotoxins. Okay, these are the semi-synthetic derivative of the epipodophylotoxin and isomer of the naturally occurring agent called the podophylotoxin. Now both of the agents act as a topoisomerase poisons. Now again I have mentioned what is the topoisomerase poison. So topoisomerases, when the leaving the supercoiling of the DNA, they have to bind with the DNA and form a DNA DNA topoisomerase complex, which is also known as the cleavage complex. Okay, why it is known as the cleavage complex? Because the topoisomerase enzyme, they have to cut the DNA strand in one position and then they attach to it. Okay, and they do how they do it already I have mentioned. Okay, so to the topoisomerase one, they cut on the DNA one strand of the DNA, and the topoisomerase two, they cut on the different positions. Okay, so this DNA topoisomerase cleavage complex, so the agents which stabilizes this cleavage complexes are known as the poison. DNA strand breakage by the class of the molecules are thought to occur by free radical process involving 4' phenolic group, but this 4' phenolic group, or what is proposed to happen is that this 4' phenolic group forms a semiquinone radical which generates again reactive oxygen species. Okay, evidence of this phenolic OH group forms a semiquinone radical structure which and the radical mechanism is followed here. Evidence for this comes from if you remove this OH, okay, or if you methylate this OH group, this compound becomes inactive. So presence of the glycoside sugar moiety here, it increases the ability of the molecule to induce the DNA breaks. Okay, so remember this the topoisomerase acts as a non-intercalating topoisomerase poison. Okay, so these are the non-intercalating topoisomerase poison. This para OH group is important because it gives rise to a semiquinone structure and radical formation happens, which gives rise to the reactive oxygen species, which is the mechanism of action. And how you can prove this mechanism of action? If you remove this OH or methylate this OH with the OMe group, the compound becomes inactive. Okay. So this the number B was the correct answer. Etoposide is the non-intercalating topoisomerase poison. Now let me go to the next problem. Problem number 9 says which of the following is not a catalytic inhibitor of topoisomerase? Your options are number A dextrazosane, number B aclarubicin, number C novobiocin, and number D irinotecan. Which of the following is not a catalytic inhibitor of topoisomerase? Dextrarosexane, number B aclarubicin, number C novobiocin, and number D irinotecan. Which of the following is not a catalytic inhibitor of topoisomerase? Read all the options carefully and think whatever we have learned in today's class so far. I have discussed it already regarding this, so I think you should be able to answer this question correctly. Which of the following is not a catalytic inhibitor of topoisomerase? I will wait for a minute, then I will check the chat box. A, B, C, or D, let me know which of the following will be the correct answer. Which of the following is not a catalytic inhibitor of topoisomerase? Which of the following is not a catalytic inhibitor of topoisomerase? I think I should check the chat box. Okay, number A, number B. So I have received two answers. One is saying number A is correct and another is saying number B is correct. Let me check which of the following is the correct answer. So the correct answer is number D, irinotecan. Already I have told you that I think I have already discussed it indirectly that camptothecin and the analogs of camptothecin, they act as the topoisomerase poison, not the catalytic inhibitor. Okay, so that's why the correct answer is number D, irinotecan. So let us first have a clear understanding what is the topoisomerase poison and what is the catalytic inhibitor of topoisomerase. So, in oncology, topoisomerase inhibitor is an umbrella term for the drugs that disrupt the topoisomerase enzyme. Within this group, there are critical distinction between the topoisomerase poison and catalytic inhibitor. Okay, the topoisomerase poison which actively create DNA damage and catalytic inhibitor that simply stop the enzyme from working. So the topoisomerase poison, what are the features? The primary goal to turn the enzyme into a weapon that breaks the DNA, and catalytic inhibitor to stop the enzyme from performing its normal DNA untangling work. Mechanism stabilizes the cleavage complex where the enzyme has already cut the DNA, preventing it from being resealed. Okay, and the catalytic inhibitor, they interferes the early and late stages of the enzyme cycle like DNA binding, ATP binding, or ATP hydrolysis. DNA damage causes permanent lethal double strand break or single strand breaks. Catalytic inhibitor does not typically induce direct DNA breaks. Cytotoxicity, the topoisomerase poisons are highly cytotoxic. Effectiveness often increase the higher enzyme levels. The catalytic inhibitor generally less cytotoxic can cause resistance if they prevent a poison from binding. Clinical use, most standard chemotherapies, for example, etoposide, doxorubicin, irinotecan, they are the examples of topoisomerase poison. Limited clinical use for the inhibitors. Some are used to reduce toxicity or treat rare conditions. For example, this dextrosozan or aclarubicin. Okay, so basically topoisomerase are the class of enzyme which this relieves the supercoiling of the DNA. So how does it works? If you go through the particular mechanism of action of topoisomerase that I have discussed earlier, that it creates a break on the DNA and binds to it, and then it actually moves the strand rotation and relaxes the supercoiling. Okay, but this topoisomerase poisons, what they do, they basically binds with this DNA topoisomerase complex, and this religation cannot happen, and this traps this cleavage complexes. Okay, so that's why these are very lethal. But the catalytic inhibitor, they do not bind with the DNA enzyme complex. They actually binds with the enzyme and distorts the structure so that the enzyme now cannot bind to the DNA. But in oncology, topoisomerase poisons are most important, more important than the catalytic inhibitors, because they are highly cytotoxic, and the effectiveness increases with the higher enzyme level. Okay, so these are the differences with the topoisomerase inhibitors, about the topoisomerase catalytic inhibitor and poison. Now, problem number 10, which of the following statements is true regarding cisplatin? Your options are: number A, this disrupts the base pairing of the adenine and guanine. Number B, interstrand cross-linking of DNA results from the action of the drug. Number C, base pairing of cytosine and guanine is disrupted by this. Number D, it has a tetrahedral geometry. Which of the following statement is true regarding cisplatin? Again, read all the options carefully and let me know A, B, C, or D. Which of the following you think is the true statement regarding the cisplatin? Which of the following statements is true regarding cisplatin? Number A, this disrupts the base pairing of adenine and guanine. Number B, interstrand cross-linking of DNA results from the action of the drug. Number C, base pairing of cytosine and guanine is disrupted by this. And number D, it has a tetrahedral geometry. Which of the following statements is true? Let me know A, B, C, or D. What of the you think is the correct answer? Let me check the chat box. Let's see if there is any answer for problem number 10. Okay, I have got one answer. Okay, number D, number A, number B. So I have received number three different options as the answer. Let me check which of the following is the correct answer. Which of the following statements is true regarding the cisplatin? So the statement which is true regarding cisplatin will be, it is number C. Base pairing of cytosine and guanine is disrupted by this. So whenever there is a true false kind of question, please read the question and options very carefully. Sometimes the confusion might happen. Okay. So the statement which is true is the number C. Base pairing of cytosine and guanine is disrupted by cisplatin. So this is the structure of cisplatin and transplatin. So it has a square, it has a it does not have a tetrahedral geometry. It had a square planar geometry. Okay, as you can see, all the groups are the same plane. So this is a square planar geometry. So number D is the incorrect option. Number A, which says that it disrupts the base pairing of adenine and guanine, it is also false. Okay, adenine and guanine at a first place does not form a base pair. Adenine bonds with the thymine and guanine adds with bonds with cytosine. So this is also false. It also number B, interstrand cross-linking happens. No, actually intrastrand cross-linking happens. Okay, so this is the so first in the plasma membrane is the plasma membrane. So outside it, the chloride ion concentration is very high in the blood. So this is the active, this is the form of the cisplatin. So when it goes to inside the cell where the chloride ion concentration is low, so it this is this for chlorides are replaced by this water molecules. Okay, and this is the active form of the cisplatin. Then it binds with the N7 of the guanine base pair, and okay, and this is the adenine guanine DNA base pair. It binds with the N7 of the guanine and it forms the intrasand addict with the as the major product. Okay, this is so this is very important. Remember this, that this cisplatin, it forms 1,2-D GPG intrastrand addict as the major product, not the interstand. Okay, so after, so this now gives rise to the disruption of the DNA double helix. Okay, so now read the options again carefully. Number A, it disrupts the base pairing of adenine guanine. This is false. Number B, interstrand cross-linking happens. This is also false because interstrand adduct is formed. Number C, base pairing of cytosine and guanine is disrupted. This is true. And number D, it has a tetrahedral geometry. It is also false. Cisplatin has a square pyramid, a square planar geometry. Okay. So number C was the correct answer. Now I will go to the next problem, problem number 11, which says which of the following reaction is involved in the method of action of calcimicine. Your options are number A, Bally-Silman reaction, number B, Suzuki coupling reaction, number C, Diels-Alder reaction, and number D, Boltzmann cyclization reaction. Which of the following reaction is involved in the method of action or mechanism of action of calcimicine? Bally-Silman reaction, Suzuki coupling reaction, Diels-Alder reaction, or Boltzmann cyclization reaction. Let me know A, B, C, or D. Which of the following is the correct answer for problem number 11? I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D. Which of the following will be the correct answer for problem number 11? Which reaction is involved in the method of action of calcimicine? Bally-Silman reaction, Suzuki coupling reaction, Diels-Alder reaction, or Boltzmann cyclization reaction? Let me check the chat box. Let's see if there is any answer for problem number 11. Number D, number A. Okay, so I have received two answers. One is saying number A, another is saying number D. Let me check which of the following is the correct answer, number A or number D. So the correct answer will be, it is number D. Bergman cyclization reaction is involved in the mechanism of action of the calcimicine. Okay. So first let us understand what is the Boltzmann cyclization reaction. So here we have an indiene structure where the triple bond is followed by a conjugation with a double bond, and then there is also conjugation with the triple bond. So this is known as the indiene, okay, structure. So after the application of the heat or light, it forms a 1,4 benzene diradical, okay, or benzyl didyl radical, which is the goes through the mechanism of action like this. It abstracts the hydrogen from here, and also radical is formed, and this type of cyclization happens. So from this we get this. So this is known as the Bergman cyclization reaction, which goes through the radical pathway. So this is the compound, or this is the drug which is known as the calicimicine. So this, as you can see, there is an indiene structure. The triple bond is single bond, then double bond, then single bond, then triple bond. Okay, and here also there is a sugar background, and there is a sulfur group. So first the nucleophile, it attacks on this position, and we have this nucleophilic thiol group here present. This thiol lone pair attacks on this position, and here there is a rotation of the double bond which happens, and this structure is formed where the sulfur creates a cyclic structure. Okay, so this is the form A, and this is the form B. From here we have this Bergman cyclization on this indiene structure, and due to the Bergman cyclization, we have a cyclic structure here where, with the 1,4 benzene diradicals. Okay, so these benzene diradicals react with the DNA, and they create the double strand DNA double strand cleavage. Okay, in presence of the oxygen. So this is the one kind of mechanism which is known as the chain cutting mechanism, and this calcimicin also known as the chain cutter or chain cutting reagent. Okay, so chain cutters, they cut the DNA strand and prevent the enzyme DNA ligase from repairing the damage. They appear to act by creating radicals on the DNA structure. These radicals react with the oxygen to form a peroxide species and DNA chain fragments. Natrichmycin gamma 1 is an example of the chain cutter. This compound binds to the minor group for the DNA and cuts the DNA chain. The reaction starts with a nucleophile attacking to the trisulfide group. The thiol, which is frayed, then undergoes an intermolecular Michael addition with the reactive alpha beta unsaturated ketone. The resulting intermediate then cycloaromatizes the reaction known as the Bergman cyclization to produce an aromatic diradical species which snatches the two hydrogens from DNA, and as a result the DNA becomes diradical. Reaction with oxygen then leads to the chain cutting. This is the mechanism of action of the chain cutter reagents. Okay. So remember, calcimicin is an example of chain cutter. This is the mechanism of action where the main one of the very crucial step is the Bergman cyclization, which creates the biradical. This biradical takes the hydrogens from the DNA by creating DNA radical, which as a result which reacts creates the DNA strand cleavage. Okay. Now I will go to the next problem. Problem number 12 says acyclovir, the structure is this. Okay, the acyclovir is an inhibitor of DNA polymerase selectively in virus infected cell. This is because number A, acyclovir is first converted to the monophosphate only by the viral thymidine kinase. Number B, acyclovir does not enter infected cells. Number C, acyclovir first converted to monophosphate only by host thymidine kinase. Number D, acyclovir triphosphate is not accepted by the host DNA polymerase. Which of the following is the correct answer for problem number 12? Number A, acyclovir first converted to monophosphate only by viral thymidine kinase. Number B, acyclovir does not enter infected cells. Number C, acyclovir is first converted to monophosphate only by host thymidine kinase. And number D, acyclovir triphosphate is not accepted by the host DNA polymerase. Which of the following will be the correct answer for problem number 12? Let me wait for a minute or two, then I will check the chat box. Regarding the acyclovir, we will be discussing again in detail in when we will be discussing regarding the antiviral drugs. Okay. Since it is related because it is an inhibitor of DNA polymerase, I thought to give the question here today. Let me check the chat box. Let's see if there is any answers for problem number 12. Okay, I have received three answers, one option A and two option C. So two different options as an answer, number A and number C. Let me check which of the following is the correct answer. So the correct answer will be it is number A. Acyclovir first converted to monophosphate only by viral thymidine kinase. Okay, that is why it is selectively used as the used in the virus infected cell to inhibit the DNA polymerase enzyme. So these are also known as the chain terminators. Okay, normal replication occurs when a normal replication occurs when the triphosphate reacts with the 3' end and forms a phosphoriester bond that catalyzes the DNA polymerase. Acyclovir is an important antiviral drug which was discovered in 1970s and acts as a chain terminator, satisfying all the three requirements of the chain terminators. Okay. So what are these requirements? First, it contains a guanine base, which means it can base pair to cytosine moieties on the template chain. Okay, so as you can see here, there is a guanine base, and with it, it has a acyclovir prodrug. It is here. Acyclovir triphosphate is the active drug. Okay, and this is the secondly, although it does not contain triphosphate group, it is added when the molecule in virally infected cell. Thirdly, the sugar unit is incomplete and lacks the required OH group and normally present in the 3' position. And the nucleic acid chain does not get extended. So, particularly this acts as a chain terminator, okay, because it acts as a growing chain, but it cannot lead to the particular. So, this is the DNA template. This is a growing chain in the DNA replication. Now, the drug, which is acyclovir, it binds with the cytosine because it has a structure which is same with the guanine. So it acts as a guanine and binds with the cytosine to create a base pair, the cytosine guanine hydrogen bonding. But then the chain now the next step is the cannot happen because it does not the sugar unit is incomplete because it does not have a required OH group in the 3' position. Since it is not present, so it cannot normally present in the 3' position. So now the chain cannot get exceeded and it acts as a chain terminator. So it is a inhibitor of the DNA polymerase enzyme, and especially it happens in the virus infected cell, not the host cell, because only in case of the virus cell, the inactive form of the acyclovir, it goes to the active form by as a acyclovir triphosphate. Okay, and this reaction only is done, it is possible by the viral thymidine kinase enzyme. Okay, not by the host. That is why it is a selective DNA polymerase inhibitor for the virus infected cell, not the human cells. Okay. Now I will go to the next problem. Problem number 13 says deprivation of adequate oxygen in the tissues is termed as number A hypoxia, number B ischemia, number C homeostasis, number D metabolism. Which of the following is the correct answer? Deprivation of adequate oxygen in tissues is termed as which of the following? Number A hypoxia, number B ischemia, number C homeostasis, and number D metabolism. Let me wait for a minute or two, then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer. Deprivation of adequate oxygen in tissues is termed as which of the following? Hypoxia, ischemia, homeostasis, or metabolism? Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 13, which is the correct term. So deprivation of adequate oxygen in the tissues is termed as which of the following? Let me check if there is any answers in the chat box. Okay, I have got several answers and all are saying no. I have one C and other all are A. So I have majorly received A as the answer with a C also. So let me check which of the following will be the correct answer. So the correct answer will be it is A. Hypoxia is the term which is used to define the deprivation of adequate oxygen in the tissues. So hypoxia is a condition in which the body or a region of the body is deprived of the adequate oxygen supply at the tissue level. Hypoxia may be classified as either generalized affecting the whole body or local affecting the region of the body. So what are the types of hypoxia? So there is a different types of hypoxia is present. We have the hypoxic hypoxia, which is the most common type caused by the lack of oxygen reaching to the lungs. Common causes is include the high altitude like low atmospheric pressure, drowning or lung diseases like COPD or asthma. Then anemic or hypemic hypoxia occurs when the blood's oxygen carrying capacity is reduced even if the lungs are healthy. Causes include lungs are healthy. Causes include anemia, severe blood loss, or carbon monoxide poisoning. Then we have the transient hypoxia, results from the poor blood circulation, preventing oxygen from reaching to the tissues. This can be general, like heart failure, shock, or local, like blood clot or Raynaud syndrome. Histotoxic hypoxia, tissues are adequately supplied with oxygen but cannot use it due to the cellular poisoning, such as from the cyanide and certain narcotics. Metabolic hypoxia happens when oxygen demand exceeds the body supply during the high metabolic status like sepsis or intense exercise. Okay. So basically, this is the hypoxia and this is the tumor regression. So now what? So when hypoxia is affects basically where the hypoxic environment I mean forms. Let me give you an example. So in cases of solid tumors, when the tumors are solid inside the cells which are inside that solid tumor, they actually experiences this hypoxic environment. Okay, and so so this is one the example. So like where this hypoxia can happen, and these are the regulators, downstream mediators, cellular effects, and the pathological outcomes for the hypoxia. So remember what is the hypoxia and what are the different types of hypoxia. Okay. Now I will go to the next problem. Problem number 14 says, which of the following is not an example of the T cell? Your options are number A CD4 plus cell, number B killer cell, number C plasma cell, and number D suppressor cell. Which one of the following is not an example of T cell? CD4 plus cell, killer cell, plasma cell, or suppressor cell? Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer for problem number 14. Which one of the following is not an example of T cells? CD4 plus cell, killer cell, plasma cell or suppressor cell? Okay, let me check the chat box. Let's see if there is an there will be answers I know. Let me check. Okay, number C, number B, number C. So I have received two number C and one number B as the answer. Let's see what is the correct answer. So the correct answer will be the one which is not an example of T cell. It is number C. Plasma cells are not is the example of the T cell. Now, first, now let us understand what are the T cell and what are the B cell, and let us have a quick comparison between two. So, B cell they attack the invaders outside the cells, and T cells they attack the infected cells. So, there is a subtle difference between these two. Okay, not subtle. There is a huge difference between these two. So, the B cells they will attack the invaders outside the cells. Okay, but the T cell they itself will attack the infected cell and kill them. So T cells they specialize in identifying and destroying the cells that have already been compromised by viruses or cancer and coordinating this overall immune response. Because there are different T cells like helper T cell like CD4. They act as a coordinator of the immune system. They release the cytokines to activate other cells including B cells and cytotoxic T cell, cytotoxic or killer T cell like CD8 plus. They directly attack and kill the infected and cancerous cells by releasing toxic granules that produce the apoptosis or cell suicide. Regulatory or suppressor T cell, they help to wind down the immune response. Once an infection is cleared, they prevent the body from attacking its own healthy tissues. Memory T cell or long-lived cells that persist after an infection to ensure the rapid response if the same pathogen returns. Then B cells are essential for antibody factories that target the pathogens circulating freely from the body like plasma cell. The effector form of the B cell once activated, they secrete the massive amount of antibodies and neutralize the toxin and tag the bacteria and viruses by destruction of the by other immune cells. Memory B cells similar to memory T cell they stay in the body for years and decades, enabling the immune system to produce antibodies almost immediately upon the re-exposure. So what are the differences between the T cell and B cell? So T lymphocyte or T cell, the maturation site is the thymus gland, whereas the B cell is the bone marrow. The immune type, the cell-mediated immunity is from the T lymphocyte. They attack the infected cell. B lymphocyte humoral immunity produces the antibody. Target recognition T cells by antigens present on the MHC molecules and free floating of antigens of the blood and lymph in case of the B cell. Surface receptor T cell receptors and B cell receptors in case of the B cell. Lifespan generally long-lived T cells and the B cells are short-lived. So CD4 plus cell, killer cell, suppressor cell, all are the example for the T cell. But the memory cells are sorry, the plasma cells are the examples of the B cells. Okay, so these are the about the T cell and B cell. Now let me go to the next problem. Problem number 15 says which of the following is known as the antigenic determinant? Your options are number A hapten, number B epitope, number C enzymes, and number D paratope. Which of the following is known as the antigenic determinant? Hapten, epitope, enzymes, or paratope? Again, after 1 minute, I will check the chat box. Let me know A, B, C, or D. Which of the following is the correct answer? Which of the following is known as the antigenic determinant? Hapten, epitope, enzymes, or paratope? Which of the following is known as the antigenic determinant? Hapten, epitope, enzymes, or paratope? Let me check the chat box. Let's see what are the answer that I have received. Okay, number B, so all our answers are with number B. So let me check if number B is the actual correct answer or not. So the correct answer, the one which is known as the antigenic determinant, will be yes, it's number B, epitope. So So in case of this antigen, we have the some structures which known as the epitope, and on the antibody we have a location which known as the paratope. So this and this epitopes on antigen, these are known as the antigenic determinant. So an antigenic determinant is commonly known as epitope, is a specific portion of an antigen that is recognized and bound to the immune system. It is the smallest unit of antigen that possesses antigenicity, means it can trigger a targeted immune response. Key characteristics: size. They are typically small molecule fragment, often consisting on 5 to 8 amino acid residues in proteins or 1 to 2 sugar units in carbohydrates. Specificity: the immune system produces unique antibodies or T cell receptors that fit the specific shape and chemical configuration of the determinant, much like a lock and key. Multiplicity: a single large antigen like virus or bacterium can have hundreds of the different antigenic determinants on its surface, allowing the immune system to attack it from multiple angles simultaneously. Main types of the antigenic determinants: linear or sequential determinant. They consist of continuous sequence of amino acid along with the side chain, along with the protein chain. T cell primarily recognizes these linear fragments after an antigen has been broken down or processed by the body. Conformation and discontinuous determinant. These are formed by the different parts of the protein sequence are brought together in the space by the protein's 3D folding. B cells and antibodies often target this surface shape. If protein is denatured or unfold, this determinant may be destroyed. Okay, so these are the main type of the antigenic determinants. So this is the example. This is the linear epitope, okay, or the continuous epitope. The amino acid residues are adjacent, so the polypeptide chain, and this is the discontinuous, okay, or conformational, were created from the amino acid residue located in the different parts of the polypeptide chain. So these are the these are the different types, and these are the figures. And remember that the portion of the antibody where the antigen binds is known as the paratope, and the portion of antigen which acts is recognized by the immune system is known as the epitope, and epitope is the antigenic determinant. Okay, so yes, number B was the correct answer. Now problem number 16 says, which one of the following statement is false for the primary metabolites? Your options are: number A, these are the part of the basic molecular structure of organism. Number B, vitamins are examples of primary metabolites. Number C, they are not directly involved in the growth, development and reproduction. And number D, these are produced in large quantities. Which of the following statements is false for the primary metabolites? Again, I will repeat. Number A, these are a part of the basic molecular structure of an organism. Number B, vitamins are examples of primary metabolites. Number C, they are not directly involved in growth, development and reproduction. And number D, these are produced in large quantities. Which of the following statements is false regarding the primary metabolites? Read the options carefully and then let me know A, B, C or D, which of the following is the correct answer. Which of the following statement is false regarding primary metabolites? Read all the options carefully and then let me know. Number A, these are a part of the basic molecular structure of organism. Number B, vitamins are examples of primary metabolites. Number C, they are not directly involved in the growth, development, and reproduction. Number D, these are produced in large quantities. Let me check the chat box now. Okay. Number C, number C. So I have received three answers. All are saying number C is the statement which is false. Let me check if it is so. So the statement which is false is number C. They are not directly involved in growth, development, reproduction. This is the false statement regarding the primary metabolites. So, primary metabolites are the chemical compounds produced during the growth and development processes. They are also involved in the primary metabolic processes like respiration and photosynthesis. The primary metabolites are formed in the growth phase. They maintain a physiological function of the body and are known as the central metabolites. They are the intermediate products of the anabolic metabolism, which are used by the cells for the formation of the essential macromolecules. Amino acid, vitamin, organic acids are the some of the primary metabolites produced industrially. Alcohol is a major primary metabolic produced in the large scale or industrially. Primary metabolites versus secondary metabolites. Primary metabolites perform physiological functions in the body like intrinsic functions. Secondary metabolites are derivatives of the primary metabolites. Primary metabolites are directly involved in the growth, development, and reproduction, whereas the secondary metabolites play ecological functions. Primary metabolites form during the growth phase due to the energy metabolism and produced in large quantities, and extraction is easy. Secondary metabolites formed near the stationary phase of the growth, produced in small quantities, and the extraction is difficult. Primary metabolites are small in all plants but unique. The secondary ones are the unique in different plant species. Primary metabolites are a part of the basic molecular structure of an organism. For example, ethanol, lactic acid, nucleotide, vitamins. Secondary metabolites are not a part of the basic molecular structure of an organism. For example, pigment, antibiotics, and drugs are the examples. Okay. So number C was the correct answer. Now I will go to the next problem. Problem number 17 said the correct set of three coenzyme involved in the biosynthesis of acetyl-CoA are number A TPP, FAD, NADH, number B TPP, lipoic acid and FAD, number C FAD, NAD plus and lipoic acid, and number D lipoic acid, NADH and TPP. Which of the following will be the correct set for the of coenzyme involved in the biosynthesis of acetyl-CoA? Number A TPP, FAD, NADH. Number B TPP, lipoic acid and FAD. Number C FAD, NAD plus and lipoic acid. And number D lipoic acid, NADH and TPP. Which will be the correct set of three coenzymes involved in the biosynthesis of acetyl-CoA? Let me know A, B, C or D which of the following you think is the correct answer. I will wait for another minute and then I will check the chat box. Let me check the chat box. Let's see if there are any answers for problem number 17. Okay. Number C, number C, number C. So I think I have received one number A and all other are number C. So let's see which of the following will be the correct answer. So the correct answer is number B. TPP, lipoic acid and FAD is the correct set of the coenzymes which are involved in the biosynthesis of acetyl-CoA. So the conversion of pyruvate to acetyl-CoA, it takes up a few, coenzymes. TPP, lipoate or lipoic acid, and FAD is the correct set of coenzymes that are involved. Okay. So the net reaction oxidative decarboxylation of the pyruvate, and then this is the first two carbons of the glucose to be fully oxidized. Okay, they catalyzed by PDH. They require 5 coenzyme. TPP, lipoate, and FAD are the prosthetic groups of the core coenzymes. NAD plus and NAD plus is there. NAD plus and acid this this coenzyme and A. So these are the co-substrate. Okay, so they acts as a co-substrate in case of the in case this of the reaction. So NAD plus is not does not act as a coenzyme, but it acts as a co-substrate. Okay, so this is the pyruvate and this is the acetyl CoA. The net reaction uses this enzyme pyruvate decarboxygenase, coenzymes TPP, lipoate, FAD, and co-substrate is this NAD plus and coethiol. Okay, so the correct answer was number B. Now, problem number 18 says, for which phase of cell cycle anthracycline anti-cancer agents are toxic? Your options are number A S, number B M, number C G1, and number D G2. For which phase of cell cycle the anthracycline anti-cancer agents are toxic? S, M, G1 or G2? For which phase of cell cycle anthracycline anti-cancer drugs are toxic? Phase S phase, M phase, G1 phase or G2 phase? For which phase of cell cycle this anthracycline anti-cancer drugs are toxic? Let me know A, B, C or D which of the following will be the correct answer for problem number 18. For which phase of cell cycle anthracycline anti-cancer drugs are toxic? S phase, M phase, G1 phase or G2 phase? Let me check the chat box. Let's see if there are any answers for this problem number 18. Number B, number A. So I have received two answers. One is saying number A, another is saying number B. Let's see which of the following will be the correct answer, number A or number B. So the correct answer is number A. For S phase of the cell cycle, this anthracycline anti-cancer drugs are toxic. So These anthracyclines are they can work as antibiotics, or also they can work as the anti-cancer agents. The details of this we will be discussing when we go through our I think week 10 or week 11 course content, when we are discussing about this about antibiotics and anti-cancer in detail. So remember the anthracycline class of the anti-cancer agent, for example, doxorubicin and darubicin are there. So they acts as a intercalators. We have already discussed that they acts as a intercalator. Okay, and for as the intercalating, they have this cyclic structure which slides to the double helix and form the van der Waal interaction with the base pairs, and also we have this amino group, the side of the amino sugar backbone, which interacts with this particular sugar phosphate backbone. So this is the DNA doxorubicin complex. So the properties anthracyclines are the tetracycline rings with the sugar donosamine. They are the DNA intercalating agent that block the synthesis of DNA and RNA. These agents primarily toxic during the S phase of the cell cycle. Since it block the biosynthesis of DNA, and in the cell cycle, during the S phase, this happens. So basically, these agents are inhibits this S phase of the cell cycle. Doxorubicin is probably the most important anti-cancer drug. Already, I have discussed it because it's a relatively broad spectrum of the activity. So intercalation prevents the normal chain of the topoisomerase 2. This also I have already discussed. The mechanism of topoisomerase 2 involve the formation of DNA enzyme complex, where the enzyme is covalently linked to the DNA. When doxorubicin is intercalated into DNA, it stabilizes the DNA enzyme complex and stalls the process. Okay. So remember, this anthracycline class of anti-cancer drugs, they are toxic for the S phase of the cell cycle. Okay. Let me just highlight it so that you do not miss this portion. And already I have discussed the mechanism of action of the doxorubicin and other anthracyclines in detail in our today's class itself. Okay. So problem number 19 says the following structure is used for the treatment of the brain tumor. What is this structure called? Your options are number A carmustine, number B lomustine, number C streptozotocin, and number D cyclophosphamide. What is the name of this structure? Carmustine, lomustine, streptozotocin, or cyclophosphamide? This is used in the treatment of the brain cancer. Okay. Let me know what is the name of this structure: carmustine, lomustine, streptozotocin, or cyclophosphamide. I will wait for a minute or two, then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer. What is the structure called? This is a drug which is used in the treatment of the brain tumor. What is the structure of this drug called? Carmustine, lomustine, streptozotocin, or cyclophosphamide? The following structure is used for the treatment of brain tumor. What is this structure called? Your options are number A carmustine, number B lomustine, number C streptozotocin, and number D cyclophosphamide. Let me check the chat box to see if there is any answer for problem number 19. Number D, number B, number A, number, okay, I have received several answers. Number B, A, number B, B, number D. So I have received three different options, number B, A, and number D as the answer. Okay, let me check which of the following will be the correct answer. So the correct answer will be It is number B. Lomustine is the name of this particular drug. Okay, so lomustine is an alkylating nitrosourea compound. Alkylating agents already I have discussed, like cyclophosphoramide is an anti-alkylating agent which I have already discussed. So this is the also an alkylating agent. This drug is lomustine. It is an alkylating nitrosourea compound used in chemotherapy. It is closely related to the semustine and is the same family of streptozotocin. It is highly lipid soluble drug, thus it crosses the blood-brain barrier. Since it is a lipid-soluble drug, it can cross the blood-brain barrier. This property makes it ideal for treating the brain tumor, which is its primary use, although it is also used to treat the Hodgkin lymphoma as the second-line option. So this is the structure of carmustine, which works by slowing or stopping the cancer cells. This is the structure of streptozotocin antibiotic that produces the pancreatic islet beta cell destruction. And this is the structure of cyclophosphoramide, which I have discussed. It is used to treat the cancer of the ovary, breast, blood, and the lymph system. Okay, so the main importance of the lomustine is that is the high lipophilicity property. So okay, since it has a high lipophilicity, it is a highly soluble in lipids, so it can cross the blood-brain barrier easily. So whenever the drug is required to reach the brain, it must cross the blood-brain barrier. Otherwise, it is not possible for a drug to reach the brain. That is why lomustine is ideal for treatment of brain tumor, because it does the same by penetrating the blood-brain barrier because of this lipophilicity. Okay. Now, the last question of today's session, it says which of the following is an example of the secondary metabolite? Your options are number A, caffeine, number B, rubber, number C, glucoinolate, and number D, all of the above. Which of the following is an example of the secondary metabolite? Caffeine, rubber, glucoinolates, or all of the above? Again, I will wait for a minute or two and then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer. Which of the following is an example of secondary metabolite? Caffeine, rubber, glucoinolates, or all of the above? This is the last question of today's session, so I want all of you to participate and answer. Which of the following is an example of secondary metabolite? Caffeine, rubber, glucoinolates, or all of the above? Let me know. Let me check the chat box. Let's see if there is an answer for this problem. Okay, number B, number D, number B, number D, number D. So I have received almost all number D and one I have received as number B. Now let me check which of the following will be the correct answer. So the correct answer will be, it is number D. All of the above are the examples of the secondary metabolites. So secondary metabolites I have already discussed. They are also known as the specialized metabolites, or organic compounds produced by organism, including the plants, fungi and bacteria, that are not directly essential for basic growth or development or reproduction. Instead, they play a critical role in organisms' long-term survival, environmental adaptation, and ecological interaction. Key characteristic: non-essentiality. Absence of this metabolite does not cause the immediate death, unlike the primary metabolites, for example, amino acids and sugar, but significantly reduces the organism's fitness in the wild. Species specificity, they are often unique to the specific species or families, making them useful marker for biological classification. Late phase production, in many microbes, they are typically synthesized during the stationary phase or growth phase, idiophase, and often triggered by stress and nutrient depletion. Major classes of secondary metabolites based on their chemical structure and biosynthetic origin, they are generally divided into four primary groups. For group 1, terpenes or terpenoid, key characteristic composed of 5 carbon terpene units and larger dimose divert class. For example, menthol, rubber, artemisinin, carotenoids are the examples of terpenes. This artemisinin is an antimalarial drug. Phenolics, compounds with an aromatic ring and one or more hexyl groups. For examples are flavonoid or pigment, tannin, resveratrol. They are found in grapes and lignin. Alkaloid, nitrogen-containing heterocyclic compound often have a potent physiological effects on animal. For example, morphine, caffeine, nicotine, quinine, and cocaine. And nitrogen or sulfur-containing non-alkaloid compound, including the specific defense molecule. For example, glucoinolate found in mustard and broccoli. Some cyanogenic glycosides. These are the examples of the secondary micrometabolites. So this was the last question of today's session. So today, majorly we have discussed about various type of reagent or various classes and of drugs that interact with the nucleic acids and acts as an anti-cancer drug. We have discussed about the majorly about the topoisomerase poisons, topoisomerase enzyme inhibitor. We have discussed about the intercalating agents, alkylating agents, chain cutter, chain terminator, and also we have discussed a little bit about the biological aspects like the metabolites, the primary, secondary ones. We have discussed about the T cell and hypoxia and all these things. So the topics are very crucial, and please go through the slides once I upload. Please go through the slides very carefully and then let me know. Secondary metabolites pharmacology. So the part of the secondary metabolites, there it is in your syllabus. If you go through the syllabus, you will find that it is a part of the biological chemistry, the primary and metabolites, secondary metabolites. Not in very detail, but a very little, just brief discussions or brief introductions about this primary and secondary metabolites. Okay, that is why I have discussed it a little bit. So this is my email ID. If you have any doubt or any query regarding today's class or any of our previous classes, or any in general queries about the course or the topics, please feel free to reach me through this email ID. Okay, so the recording I have already shown you. Let me show you one more time. You have to go to the SWAM portal of the medicinal chemistry when there is an announcement page. I think it is available all of you. You are giving the you are I think receiving the emails regarding the class reminders and also the weekly course material I mean uploading. So when you go to this SWAM portal, this course page, you will find there is a left panel here. If you go to there is a problem solving session January 2026. If you click on this portion, you will find this problem solving session recording. Once you click on this problem solving session recording, you will find a Excel sheet which contains the YouTube links for the class recordings, and also you will find a Google Drive link. This Google Drive contains all the PPTs. The PPT which I am showing you today, I will be uploading to this particular Google Drive. So all this you will find to this particular page of the SWAM portal. Okay, so you can go through this material. Okay, I think if there is any no doubt, I will close the session for today. Thank you all for joining, and I hope you have understand the topics. If you have any queries, please go through the slides. I think it will be there won't be any issues if you go through the slides properly, and also go through the book which I have already mentioned. And if you have any queries, please feel free to reach me through the email, or you can let me know in the next class also. Okay, I will close the session for today if there is no okay, the slides also I have mentioned. If you go to the particular problem solving session recording, there is a presentation folder link, which is a Google Drive link. This Google Drive is open. If you go to the Google Drive, you will find this presentation here. Okay. Just go to this this NPTEL page. You will find all the thing in the same place. Okay. The slides and also the recording. I hope it is clear where to find the slides and where to find the recording. And anything else? If there is no query, I will close the session. Thank you all for joining. And please let me know. We will meet on the next session, which will be on next Monday from 6 p.m. Okay. Thank you all. I will close the session for today.